Hello children, today I welcome you all for the fifth session of the chapter Cell of Biology. So before I start this class, I would like to have a quick recap of my previous class topics. So in the previous class, we finished the cell membrane part. We discussed the transport system of the cell membrane. We discussed about the cell wall present in the different type of cells, its components as well as its function in the cell. Then we began with the next important part of the cell, that is the nucleus. It is the brain of the cell. Why it is brain of the cell? It is controlling other cells or cells other activities. It controls the growth of the cell, and it is also playing an important role in the cell division or cellular reproduction. So we discussed the different parts of the nucleus like nuclear membrane, nuclear pore, nucleoplasm, nucleoid, chromatin fibers. Then we discussed about the location of the RNA and the protein in the nucleolus also. So today we were supposed to begin with the important component of the cell, that is chromosome. Chromosome. What are chromosomes? Chromosomes are the thread-like structures which are formed by the condensation of the chromatin during the cell division are called as chromosomes, and they are referred as hereditary vehicles. Why are they called as hereditary vehicles? Because the chromosomes, when it is formed, chromosomes are formed when the cell is in the active mode. Active mode means when it is preparing itself for the division. Division means reproduction. So that time what happens? The very fine thread-like structures, that is chromatin, condenses and forms a rod-shaped structure that is called as chromosomes. And these chromosomes are carrying the hereditary information from the parents, and during the reproduction process they are transferring these features, hereditary features, to the next generation. Okay, so in detail we will study. Chromosomes are made up of DNA and proteins. That means DNA is one of the genetic material present inside the cell. It is also called as a nucleic acid. So chromosomes are made up of nucleic acid and protein. Proteins also, there are two types of protein. One is called as structural protein and second one is called as functional protein. So the structural protein, that means give the structure. So in a chromosome, like suppose this is the chromosome, inside the chromosome the DNA is wrapped around. Since DNA is very long, so it has to be also condensed so that it will fit into the chromosome. So what happens? Who helps the DNA to fit in the chromosome? The structural protein. It is called as histones. So histones proteins are like ball-like structures. So on the ball, the DNA is wrapped and then it goes to the next histone. Then it is wrapped, then next. So in that way, the length of the DNA reduces and it is fit into the chromosome. Okay. Then another one is called as functional, which is playing an important role. That is called as non-histone proteins. Okay. Now, a chromosome consists of two similar threads called as chromatids. You can see that there are two threads in this picture. So these threads are called as chromatid. Okay, these threads are called as chromatid, and because they are together and held with a circular structure that is called as centromere, so they both are called as sister chromatids. Okay, the sister chromatids are held together by a centromere. It is looking like a bow structure. It's look like a bow structure like this, and now the bow is formed by the centromere. But in every chromosome, the location of the centromere is not same. In some, it is in the center. So the thread, it is less equally divided like in the form of X. Okay. But in that X, if it is equally, the top part of the top tail and the bottom tail are in the same length, and the centromere is just exactly in the center, that type of chromosomes are called as metacentric. If it is little above, not in the center, little on the top, then it is called as acrocentric. And then if it is in the tip of it, only the two arms are visible, then like this, like this, like this, and this two arms are visible, and here the centromere is there, so then it is called as telocentric. Okay, two chromatids are attached at the center by the centromere. On either side of the centromere, chromatids are called as arms. So these are the two arms of a chromosome. What are they? They are the two arms of the chromosome. Okay, now we go to the next picture. Number, chromosome numbers. Okay, before I go to chromosome number, I would like to discuss one thing more, that is, since I have told you that a chromosome is made up of DNA and protein. What is the role of DNA? DNA is carrying the inheritance or hereditary information. It contains all the important information which is necessary for the cell division, as well as it controls the growth of the cell and other functions of the cell. Secondly, how this genetic information is passed from the parents to progeny? That's a question. Since DNA is a long thread which is wrapped on the chromosome, some part of the DNA is carrying the functional, so it's told as functional unit. That functional unit is called as genes. So genes are located at certain location of the DNA. That genes, when combined together, father's genes and mother genes, when combined together, they form a new gene, or that gene only shows the features in the child. Okay, so remember, when the cell is dividing, then only the chromatin changes to chromosome. When the cell is not dividing, the DNA will remain in the chromatin threads only. Okay, so one thing more, when the cell needs to divide, is ready to divide, it does some preparation. This will come in the higher classes, but right now I am going to tell you, because you know that there are two types of division a cell shows. One is mitosis, other one is meiosis. So in the case of the growth, any kind of cell division, first the cell prepares itself for the division. So what preparation? Because now one cell has to be divided into two cells. So all the materials which is needed for a new cell has to be accumulated or arranged inside the parent cell. And one of the important part is the DNA. We have only one DNA in the chromosome. So that DNA has to be doubled first. So what happens? First, the parent nucleus DNA gets form a duplicate of it. That is called as DNA replication. That is called as DNA replication. That means one DNA makes another copy of itself, photocopy of itself. That is called as replication, and that process is called as transcription. That process is called as transcription. Okay, then what happens? Some of the informations are present on the RNA that has to be transferred to the DNA, and that first is DNA replication, and after the DNA replications, the information on RNA which is present, the genetic information which is present, has to be transferred into DNA, that included in the transcription. So transcription is divided in two parts. First is DNA replication. DNA makes a copy of it. Then from the RNA, the information has to be transferred to the DNA, that is called as transcription. Then that information has to be sent to the ribosomes for the formation of new proteins, new genes, like that. Okay, so this way the chromosomes helps in the cellular reproduction. Now we go to the next topic, that is chromosome number. Now different species have different number of chromosomes in their cells, but that particular species has the fixed number of chromosomes. In each species there is a fixed number of chromosomes. But as I said, The chromosomes always occur in pairs. That means if one chromosome is there, there will be another pair of it. They are always in the pair. In each pair, one chromosome is from father and one is from the mother. But in the later classes, in the higher classes, you will study that during the gamete formation or the sex cell formation, the number of chromosomes becomes half. Okay, that is what I am going to come. If in a cell there is a presence of two sets of chromosome, then that set is called as, that type of cell is called as diploid. If it is this is common, this is generally a cell gets, the cells are diploid, most of the cells are diploid. Only at the time of the gamete formation, the sex cells becomes haploid. That haploid sex cell is used for the fusion and the formation of zygote. So, at the time of the gamete formation, only one set of chromosomes is present in those cells. That is called as haploid cells. When the chromosome number is half in a cell, that type of cells will be called as haploid cells. So, in the human being, N is the number of chromosome. Since in the normal cell it is double of them, because always it is present in a pair. So definitely the number of chromosome will be double, normal cell. So generally in humans we have 46 chromosomes. That means how many pairs? 23 pairs. But like suppose I am giving you an example here. In the males, the testis is an organ, is a gonad where the sperms are forming. Okay, so sperms are what? They are the sex cells which are involved in the formation of a zygote. But that sperm will be called as a haploid cell, because when a sperm will fuse with an ovum, it will form a new cell, that is a zygote, and the zygote is a diploid cell. So only half number of chromosome comes from the father and half number of chromosomes comes from the mother. When they fuse together, they form a diploid zygote. Okay, so that half number of chromosomes will carry the genes of the father, and half number of chromosomes which is coming from the mother will carry the genes of the mother, and finally the combination of the genes takes place. And the genes is for everything, whatever you think of: eye color, hair color, skin, walking style, everything, all the features. Okay, and gradually when these genes are transferred into the zygote, slowly this chromosome only control, the nucleus only controls the growth of the the characteristics in the zygote, and finally the zygote is got all the features, combination of the features from the genes of the parents. Now, what is the role of the nucleus? We had already discussed a few. Just to sum up, nucleus contain all the genetic information, not only for the cell but for the whole organism. This I've said, not only the cell but for the formation of new cells and for giving different features in the different parts of the body. It is controlling everything. Its function as control center of the cell, as it controls the cell metabolism and cell activities. The nucleus is not only involved in passing the hereditary information, or it is in the cellular reproduction. It is controlling entire cell activities and mechanisms. The ribosomes are formed by the nucleolus. As I said, what is present in the nucleolus? RNA. RNA is responsible for the formation of ribosomes. Why? Because RNA is the genetic material or the nucleic acid which is having the codes or the information for the protein synthesis. So that protein synthesis information which is present on the RNA is transferred to the DNA for the ribosome formation, and the ribosomes are those parts of the cell which is involved in the formation of proteins. Nucleus brings about cell differentiation, by which a cell contains a specific structure and function. So nucleus only decides, forms the new cell, and they only differentiate the cell according to their function, and that gives a specific structure to the cell and the function. Division of the nucleus is essential for the cell division. As I have said, that when the cell is in the active mode and it is ready to divide, the nucleus has a very important role. How? Because the DNA is located in the nucleus, the DNA replicates, DNA takes the information from RNA, and then only it is transferred to the formation of the protein in the ribosomes. So everything is done by the nucleus. Reshuffling of chromosomes and changes in the genes produce variations. Yes, because genes are located on a specific area of the DNA. So which part of the chromosomes are going to cross with the other parent's chromosome, that changes the variation. Now suppose if this is the chromosome of the father and one gene is located here, but the mother genes is located, if when it is crossed, only the father gene was joined in the, or you can say crossed, or it came into the zygote, the mother gene has not come. So for that particular feature, only father genes are going to be dominant. So these things will come in the later class. You need not to worry for this now. Now we say that prokaryotes and eukaryotes have different in aspect of the nucleus. As I have said that in the prokaryotes, what is prokaryotes? Prokaryotes are the primitive cells in which the nucleus is not completely formed, and in those cells the nucleus is look like a very roughly formed cloud-like structure, and that is called as nucleoid. And because there is no definite nucleus in the cell, so many membrane bound organelles are also missing in the prokaryotes. Eukaryotes are well formed cells which is having a specific nucleus with a nuclear membrane, and because the nuclear membrane and a well defined nucleus is present, so that is why they are having different membrane cell organelles also, like mitochondria and endoplasmic reticulum, Golgi apparatus, and we will discuss this later. So just to differentiate between prokaryotes and eukaryotes, I am going to do this topic. Prokaryotes are the group of organisms that lack cell, nucleus or any other membrane-bound organelles. They differ from the eukaryotes because the eukaryotes are having a cell nucleus. Prokaryotes are single-celled organisms. Generally, most of the single-celled organisms, most, not all, most of the single cell organisms are prokaryotes, like bacteria that are roughly 1 micrometer in the diameter. They are very small in structure. On the other hand, a eukaryote is an organism whose cells contain complex structures inside the membrane. The defining membrane bound structure that sets eukaryotic cells apart from the prokaryotic cells. Because in a eukaryote, you will study in the further classes that there are many other parts are also present. They have a definite membrane. Because they are enclosed in a definite membrane, they have a specific function. Okay. So in a eukaryote cell, the DNA is present in the nucleus. But in the prokaryotic cell, because they don't have a nucleus, so where is the genetic material located? Inside the nucleoid, roughly formed nucleoid. Okay. Now, some more differences between the prokaryotes and eukaryotes. Prokaryotes do not have nucleus, so the genetic material houses in the nucleoid region. Prokaryote cell consists of a large DNA molecule compacted in an area called as cytoplasm, and that area is called as a nucleoid. The nucleoid region is protected and encased by a cell wall or a cell membrane, the outer layer of the cell, like human skin. Flagellum is a kind of a tail-like structure which helps the prokaryote in the mobility. Now, this is a structure of a bacterial cell. You can see that there are membrane outside. You can see, and these thread-like structures are the cilia, and this is the flagella which helps in the movement. Now the outermost line of this bacterial cell is called as a capsule. Then the inner part is called as a cytoplasm. In the cytoplasm, the ribosomes are present, and just below the capsule is the cell wall, and then below the cell wall is the plasma membrane, and there is a cloud-like structure. The thread-like structures is called as a nucleoid, which is containing a DNA. One thing new, which is, and this is also called as a fimbriae. Okay, now one thing new it is present in this prokaryote is a plasmid, that you need to understand. Plasmid is a circular DNA. Plasmid is a self-replicating circular DNA which has the ability to gather or to get the other genes from the other organism and carry it into the other organism. Like suppose they are self-replicating and circular DNA. So what happens if, like suppose this is for an example, we are talking about a bacteria. E. coli is a bacterium, and in many kind of you can say that biotechnology activities, we can take somebody's genes into other organism to get that features. Like suppose we want to make a plant a disease resistant, okay, a disease or a pest resistant. So what happens, we find that that particular plant is attacked by which type of microorganism which is making it, which is harmful that particular plant, and then we find out that what is which particular medicine or which particular feature is going to destroy that particular pest and which is not harmful for the plants. So what do we take? Suppose we take the anti pest from one organism, like suppose there is a organism which is going to kill the pest. Okay, that A organism is having a particular gene which is producing the harmful effect on the pest. So that gene we carry it and we transfer it to first E. coli, because E. coli is having what? A plastid. Plastid has an ability, plasmid has an ability to carry the other's gene to the other organism, and then it takes that genes or that feature into that organism. So if we inject the gene of any organism into the plant, plant will also develop those conditions, that harmful condition for that pest, but that harmful condition is not going to affect the plant. But when that pest will come and it's going to feed that plant, that harmful component which is produced by the genes carried by that plasmid is going to kill that pest. So this way the plasmid is helpful in carrying the genes. It is like a scooter which can carry one gene from one organism to another organism. That is why plasmid is called as a kind of a vector. Okay, so this is a special features seen in the prokaryotes. Now, just to jot down the difference between prokaryotes, you can copy it. Prokaryotes are very small. They are larger in small eukaryotes. They have a well-organized nucleus is absent. Well-organized nucleus is present with the nuclear membrane, nucleolus, DNA, everything. But in this, a single DNA molecule is present, suspended in the cytoplasm. Nucleolus is absent. Membrane-bound cell organelles are absent. Membrane-bound cell organelles are present. Typical vacuoles are absent, but typical vacuoles are present. Mitosis cannot be seen because DNA replication and all is not possible. So only the cell divides by dividing the cytoplasm. So now plant, animal cell, etc. Here bacteria, cyanobacteria, etc. Okay, so you will write down these differences in the notebook. One more thing you can write, prokaryotes have a circular DNA called as plasmid, but it is not having that type of plasmid. So we end up our class here. Take good care of yourself. Thank you so much.